Welcome to AP Biology. This video is an introduction to protein synthesis. Protein synthesis begins with an investigation performed by two scientists, Beadle and Tatum. The Beadle and Tatum experiment proved that each gene is the blueprint for the creation of a particular protein within the body. We know that a gene consists of a sequence of DNA, and that sequence of DNA codes, or is the blueprint for the creation of a very specific protein within the body. Beadle and Tatum's experiment showed this. They worked with a type of organism called Neurospora. This is a bread mold, and they looked at a particular metabolic pathway. This pathway begins with a precursor, and that precursor is eventually used to make arginine. This metabolic pathway utilizes three chemical reactions, and we know that each chemical reaction needs an enzyme. So chemical reaction 1 needs enzyme A, chemical reaction 2 needs enzyme B, chemical reaction 3 uses enzyme C. What Beadle and Tatum did was that they induced mutations in three different genes. When they mutated gene A, they discovered that this knocked out or caused enzyme A to be non-functional. When they mutated gene B, they discovered that this caused enzyme B to be non-functional. When they mutated gene C, they discovered that this caused enzyme C to be not functional. So what Beadle and Tatum said is that each gene is the blueprint for a specific enzyme within the body. This was later modified to the one gene, one protein hypothesis, and now it's been further modified to the one gene, one polypeptide hypothesis. Remember that most proteins are made up of multiple polypeptide subunits. Protein synthesis is going to take place in two chemical reactions. One reaction called transcription, or I should say one process called transcription, and a second process called translation. Transcription is going to take place within the nucleus. Translation is going to take place at the ribosomes. We have a problem here. Genes are sequences of DNA, and DNA is located within chromosomes inside the nucleus. Chromosomes are stuck inside the nucleus. However, proteins are made at the ribosomes. Both free and bound ribosomes are located outside of the nucleus. So the first process that needs to happen is we need to make essentially a photocopy of a particular gene so that that photocopy could be sent from the nucleus to the ribosome. This first process that is going to make the quote-unquote photocopy of a gene is called transcription. Transcription is going to take a gene, a sequence of DNA, and it's going to transcribe it into a sequence of what we call messenger RNA. This messenger RNA has the ability to leave the nucleus and go to the ribosome. At the ribosome, the second process called translation is going to take place. The ribosome is going to read the messenger RNA and convert it into a sequence of amino acids. We know that the sequence of amino acids is going to determine the structure or the shape of a protein, and the shape of a protein determines its function, what it's able to do. RNA is going to be an important part of protein synthesis because RNA, like it says here, is the bridge between genes and the proteins that they code for. There are multiple types of RNA that we're going to see during protein synthesis. You have mRNA, or messenger RNA, that we just saw. You have tRNA, which is transfer RNA, and you have rRNA, which is ribosomal RNA. RNA is the second type of nucleic acid. It's similar to DNA, except that there are three differences. One, RNA is single-stranded, where DNA is double-stranded. 2, RNA contains the sugar ribose, where DNA has deoxyribose, and 3, RNA has the nitrogenous base uracil instead of the nitrogenous base thymine. So it has a U instead of T. Here we see a diagram that, in my opinion, is the most important diagram to know about protein synthesis. It's a diagram that shows the overall view of what's happening. Here we see a gene. All right, we can consider this sequence right here a gene. It's a sequence of DNA that codes for a particular protein. We know that DNA is double-stranded, but only one of the 2 strands is going to be read or going to be used to make the actual protein. The strand that's used is called the template strand, and the template strand runs from 3' to 5'. The other strand is called the coding strand, and the coding strand is not the strand that's read. DNA is read in a series of triplets. It's read in a series of 3 bases. So you have 1 triplet there, you have 1 triplet here, you have 1 triplet here, you have 1 triplet here. The triplet being read is called the reading frame. This first process, called transcription, is going to happen inside the nucleus, and the DNA is going to be transcribed using an enzyme called RNA polymerase. RNA polymerase is going to read each DNA triplet, and it's going to create our sequence of messenger RNA. The messenger RNA is complementary to the DNA. Any place there's a C, there's going to be a G. Any place there's a T, there's going to be an A. The only difference is, any place that there would have been a T, you're now going to have a U. So our sequence of DNA is transcribed into a sequence of messenger RNA. Every 3 bases of RNA is called a codon. So each triplet is transcribed into a codon. We have 4 triplets right here, so we make 4 codons of messenger RNA. And again, that messenger RNA is complementary to the DNA, except that it has U instead of T. This messenger RNA, after it's been completed, has the ability to leave the nucleus, go to the ribosome, where translation will take place. During translation, the ribosome is going to read each codon, and each codon is going to correspond to a specific amino acid. How do we know what codons go with what amino acids? Here we see the dictionary of the genetic code. It tells us the different codons and the different amino acids that they code for. Every messenger RNA is going to begin with the same codon. It's always going to begin with AUG. We call this the start codon, and the start codon corresponds with the amino acid methionine. So methionine, or MET, is the first amino acid in every single protein of the body. If AUG is always the first codon, that means that TAC is always going to be the first triplet of DNA. Every messenger RNA is going to begin with the start codon. Every messenger RNA is going to end with a stop codon. If you look at the dictionary of the genetic code, you can see that there is redundancy in the genetic code, but there is no ambiguity. Redundancy means that you have multiple codons that correspond to the same amino acid. That makes sense. It's a safeguard against mutations. If a mutation occurs in the DNA that changes the codon, it doesn't necessarily change the amino acid. We would call this type of mutation that doesn't have any end result a silent mutation. But notice there's no ambiguity, meaning you never have one codon that corresponds with 2 amino acids, because then the ribosome wouldn't know which amino acid to put down. So if we look overall at this diagram here, we know that proteins control all the traits in our body. What makes one protein different from another protein is the sequence of amino acids. Sequence of amino acids determines the shape of a protein. Shape of a protein determines the function. The sequence of amino acids is determined by the sequence of bases in the codons. The sequence of codons is determined by the sequence of DNA. So ultimately, protein synthesis begins at the level of the genes. The sequence of nitrogenous bases in the genes determines the sequence of bases in the messenger RNA, which then determines the sequence of amino acids in the protein. If you just change one base of DNA, that one base DNA change, which we would call a mutation, can then change the sequence of bases in the codon, and just changing one base in the codon can change one amino acid, and changing just one amino acid can change the entire shape of a protein and cause its function to change. This concludes this video. Come on in, go. Sorry about that. I got 3 videos I have to make today, so I'm trying to knock them out. But once you start going, you have to just go.
Is protein casein